Good evening everyone and welcome to my YouTube channel. So what we are going to be looking at in A-level biology is gram staining. So why is gram staining important? Firstly, gram staining is important because it helps you identify what type of bacteria are you more like fighting against. So knowing the bacteria, the nature of the bacteria is very important in the sense that number one, what it helps you come up with is how can you more like exterminate that bacteria, or how can you eradicate or destroy or kill the bacteria. So gram staining is very important. If you are able to identify what type of bacteria are found on a colony, you can be able to effectively fight that bacteria. So the first thing about gram staining is number one. So let's say you have a colony of bacteria. Let's say this is where you are putting, you have the, let's say our yellow thing is the bacteria. The first thing that you do is fixation. So you can just do this by applying a bit of heat, just moving it here and there, so that it just gets heated, so that when you add other chemicals, they are fixed on the slide. So now, gram staining, you have to know the structures. So this is gram staining like that. So firstly, we have the peptidoglycan, which looks like that. Now the way it is, it's like this. So we have the peptidoglycan, which is the cell wall of a bacteria. So the cell wall of a bacteria determines, it is very crucial for us to identify. So firstly, the one that only has got one membrane, so this is the cell membrane. So one has got two cell membranes, the other one just has one. So this is our cell membrane. So when you look at it like this, we have this structure, the peptidoglycan, which is the cell wall. So the one that has got a thick peptidoglycan, this is what we call gram positive. The one that has got a thin cell wall, that is what we call the gram negative. So this is the gram negative. So now, you cannot necessarily identify just by looking in a microscope. You will have to use gram staining. So now, you will realize to say gram negative are very difficult to more like eradicate. This is because of their double cell membrane. So we have this cell membrane, we have another one here. So due to this, the gram negative are so difficult to more like eradicate. This is simply because we know the function of the cell membrane. It's to control what enters and leaves the cell. So let's say you are applying some antibodies. It's very difficult for the antibodies to pass, and moreover they have to pass through two cell membranes. So that's why we have the gram negative being more difficult to eradicate as compared to the gram positive. So now, the test number one, what you begin to put is, so the first chemical that we have to more like put in, so we are going to put in the crystal violet. So the crystal violet shall be added there on both of the cells. So the crystal violet, which is purple in color, so first add crystal violet. So now, one important thing that you must know is that the crystal violet will be named as your primary stain. So this is very important for your exams. You should know the primary stain is crystal violet, which is purple in color. So after you have added your primary stain, what you will begin to add is what we are going to call the mordant. So the mordant is orange in color and it is called iodine. So the iodine is added to form a complex called a mordant. So the second thing that is added, we are going to call it the mordant. So the mordant is the crystal. So we have the second thing which is added, which is our mordant. So a mordant is added and the mordant is able to create a sticky substance. So when the iodine is mixed with the crystal violet, it forms a complex which in which case the iodine acts as your mordant, which makes it to stick so that it is not easily washed away. So these terms are very important, especially for your exams and tests. So a mordant is iodine. A mordant makes the crystal violet to stick so that it is not easily washed away as we add other chemicals. So the next thing added is, so the next substance that will be added is what we call the decolorizer. So when you add your decolorizer, which is alcohol, it removes color from one of them. It does not only remove color. So the color, whenever, let's say, the iodine is mixed together with the primary stain, which we are calling the crystal violet, the color is still purple. So now, what the decolorizer does, it dries up this part is called the capsule. It dries up the capsule. It also removes the cell membrane like that. Now let's see what happens. So when the alcohol is added, which is our decolorizer, it dries up the capsule. It's more like it's a drying agent. So it dries up the capsule like that. Even the cell membrane is removed like that. So as you can see, it is removed like that. Then also the size of the cell wall is also reduced. So as you can see, this is what begins to happen. The cell wall begins to dry up like that. So this is what we have. So we only remain with a little bit of the cell wall like that, though it is not completely taken away. Then here the capsule is also removed like that. The cell wall is also more like it shrinks like that. But if we compare, because alcohol is already a liquid, it's entering, it's more like it will want to cause the substance that is here to more like move out. So now, due to the fact that the cell wall of this one is smaller as compared to this one, this complex made is removed out. It's more like it gets washed away. So what begins to happen is that this is no longer there. So after the decolorizer, the color of the gram negative is that it becomes colorless. But this one, it is more like the complex stays because of the size of the cell wall. So alcohol is a decolorizer. It dries up parts of the bacteria and also causes the complex to leave from the gram negative. Then the last substance that is added is what we are going to call the counterstain. So the counterstain, which is safranin. Safranin is used as a counterstain. So the color of this one, it's pink. So the color is pink. So this is a counterstain because it is put at the ends to see what will be the alternative color. So let us see what happens. So this is what happens. So when you put your counterstain, it more like it sticks here, it sticks here, it sticks here, it sticks here, it sticks here. Even here when you add it there, it sticks there, it sticks here, it sticks like that. So now the color now for the gram negative becomes pink, but for this one it still remains purple. The reason is simple. This is simply because of more like the crystal violet together with the complex is so dense such that it overshadows the pink color. So the bacteria is seen to be purple, then this one is seen to be pink because this is the only stain that is there after the removal of the complex made. As you can see, so we have the primary stain, which is purple in color. So it is added. So this is the bacteria. When it is added, we are going to have the purple color like that. So this applies to both gram positive and gram negative. When we have the iodine added, we still remain with the color purple. Then finally, when we add alcohol, ethyl alcohol, it more like it decolorizes the gram negative. So as you can see, these without color are the gram negative, these are the gram positive. So now at the end here, when you add your counterstain, which we are calling safranin, will, for the gram negative it will become pink, for the gram positive it will still retain purple. So let's just look at some examples. So we have, let's say, the spirochetes. This is a gram negative. They mostly cause diseases. So the spirochete is one of the bacteria that are responsible for causing syphilis. Vibrials, these are responsible for causing cholera. We can also have the gram positive. We have, these are easy to treat as compared to the gram negative because they only have one cell membrane. We have, let's say, bacillus. We also have clostridium. We also have these others. So at least note two, spirochetes, vibrials are really good examples. So we also have the one that causes gonorrhea, which we are going to call gonococcus, which is very simple because if you know already gonorrhea, you can just say gonococcus. Then we have others. So you can just at least know some examples of the gram negative and the gram positive in that matter. So thank you very much for watching my video. Hope you would like, subscribe and share for more if you want to be updated with more content. See you in the next tutorial.